Yeah, so no, thanks. It's really good to be here. And yeah, I'm just tell you a bit about, actually, so everything I'm going to show you is from the postdoc I did with Carl in Carl's lab. But I've about 5 months ago moved to Germany, and I'm in Berlin, actually, and learning German, and I'm at the Max-Delbrück-Centrum. But I'm sort of actually split between quite a few different places. And I'm actually working, my lab is here in Mitte, in the center of Berlin, because I'm part of this NeuroCure cluster of excellence, and there's a lot of new neuroscience departments come to Berlin, and we're all affiliated to different places. So yeah, I thought I'd just show you the building that we're going to be moving into. This is my lab at the moment. So I've hired postdocs to seriously get digging, and this was just after the mayor opened the site. So we hope in a couple of years we'll be there. So yeah, I'm sort of interested in what's going on in awake behaving animals, and the first person to record in the awake human was Hans Berger, and probably you know this story, but I quite like it. I think it's true. But apparently his sister or sister-in-law had a dream that he fell off a horse and broke his leg, and then actually he did, and so he phoned her, or maybe not phoned her, spoke to her, and he thought, well, you know, maybe there's some sort of telepathy going on here, and maybe these brainwaves that were sort of hypothesized at the time are somehow speaking to each other. So he started making these electroencephalogram recordings with this sort of in mind, and I think at the time was seen as a bit nuts, because he did these experiments in sort of private and didn't show anyone. He quickly realized that, in fact, telepathy wasn't really in, and in fact became the sort of father of the electroencephalogram with his amazing experiments and tight controls. He actually, this is somebody having their some of the first EEG recordings. Normally, I think he famously did it on his son, Klaus, but I don't think this is Klaus here. And these are some of the recordings that he made. So when the patient's eyes are closed, we see these large, slow amplitude oscillations, the famous alpha waves. But when they opened their eyes and started actively looking around the room, we saw a reduction in the amplitude of these oscillations, and we've got fast, small amplitude oscillations in the EEG. And here's a change from one so-called brain state to another, from the eyes open to eyes closed. So this is something that I was particularly interested in, and I chose not to work on humans, but on the mouse and the Whisker system. So yeah, you probably realize this, but there's a number of reasons why I went to work on the mouse. Yeah, as we were saying, it's a mammal. There's lots of nice molecular biology, and when I was starting my postdoc, there were really suddenly techniques available where you could really record in awake and behaving mice, and so this was very attractive for me to go and work on mice. The Whisker pathway, the anatomy is particularly well known, and also there, and perhaps most importantly, there's a very nice quantifiable behavior. So the mice actually actively move their whiskers backwards and forwards, and we can film this at very high resolution and then track the movement offline. So this is just a cartoon of well, I think it's a mouse, but some people say it's a rat, and I'm not 100% sure. But so just to show you the Whisker pathway, they normally live in dark holes, as you know, so the vision isn't so good, but they use the whiskers to build up a picture of the world, moving them backwards and forwards, and touching nearby objects. The movements of the whisker here get transduced into electrical impulses, and these pass up the infraorbital nerve into the brainstem, then to the thalamus, and finally up into the cortex. And this is where I've been making most of my recordings, in the so-called barrel cortex that processes whisker information. There's also the motor cortex for whiskers over here, and this sends signals down to motor neurons that drives whisker movement. And importantly, for later on in the talk, these nerves are separate. So the sensory nerve is separate from the motor nerve in the face of the mouse. Okay, so I've got some movies here of mice moving their whiskers. This has been slowed down a number of times, and the mouse is sitting on a platform over here, and this is in the dark with infrared light. The mouse is reaching over a gap, here, and he's trying to touch the platform over here with his whiskers. And he judges the distance of the gap using his whiskers. And you can see they're moving backwards and forwards. It's actually within a mouse around 9 times a second about they do each protraction and retraction cycle. You can see most of the time they occur in phase with each other on the left and the right side, but actually not always. Sometimes left and then right. And it's now thought actually mice can move individual whiskers to individual amounts, so they might have control over individual whiskers. To make things easier for ourselves, we actually trimmed the whiskers of the mouse, so there was only one left, so we could track it more easily. You can imagine the whiskers don't all come out at this plane. They're actually some coming up, some down. So we trimmed all of these ones and left one that sort of comes out here, and so it was just easier to film. Mice can actually do things with one whisker. So I just got a video here of a mouse doing the same gap crossing task, but at real time this time, just with one whisker attached. Here he is feeling for the gap over here, and then hop, there we go. Maybe we have it again. Yeah, just to show you that mice can do things with single whiskers. Here we've got a zoom in of the mouse also touching the platform with his single whisker. So before my experiments, I trim the whiskers, but this is only about an hour before recording. So it's not, I don't do this days and days before. So, yeah. So perhaps one of the major reasons that whiskers are such a highly used system is because of their amazing organization. And this is just a picture of a mouse. His eye is here and the nose is here. And you can really identify whiskers in every single mouse or rat. And obviously the most important whisker is this one, the C2 whisker that we've been using, but this is the one we've been identifying in each mouse. The mice, even more amazingly, Woolsey and van der Loos in the 70s discovered that in the cortex there's also a somatotopic map representing these whiskers in layer 4 of barrel cortex. And this is a slice of layer 4. This has been stained with cytochrome oxidase, and you can see these dark spots appearing. Each one of these represents a so-called barrel, and this is the input from in layer 4. In each of my experiments, I then record from a barrel that corresponded to this C2 whisker. So I'd record from this spot here, which is only a few hundred microns in diameter, but I could identify this spot in each mouse before my experiment, so I could really target the recordings to like a very tiny spot in every mouse, which is not really possible in many other cortical regions. To do that, I'll show you some of my methods that I was using. To do the targeting, I'd use this method called intrinsic imaging, and we would have a mouse anesthetized with a single whisker attached to a piezo stimulator that moved backwards and forwards, and then we have a CCD camera with red LED light shining directly on the skull, so the skull is still intact, but the skin's been removed. And you can see this is the reflected light we're measuring here. Then I start to move the whisker, and we get these dark spots appearing. So this dark spot corresponds to the barrel column that I'm stimulating. But over here you can see there's some more, but this is because our stimulator also makes a little click, and this is actually auditory cortex down here, and this is a secondary somatosensory cortex. We'd then use the blood vessel pattern on the skull. Here you can see the blood vessel pattern. So we'd use it to guide our craniotomies. So craniotomy is just a very small hole in the skull that we'd drill just before the experiments, only a few hundred microns big, as small as possible really, to improve the stability of the recordings. Then remove the dura, the membranes, and start to record. So this method actually is very reliable, and we really, from stainings of neurons, we know that we're really hitting the right spot in almost every mouse. So as I told you, my mice are awake. They're actually head restrained. They have a metal post glued to the surface of the skull, and then over a period of 2 or 3 days, or maybe up to a week sometimes, they're habituated to sitting still here, and you think they might not. And in fact, when I started, I thought they might not do this, but they do. And really, within a few sessions, they sit quite calmly for an hour or two, long enough for us to get a recording. During the recordings, we have a high-speed camera above the mouse, and they move the whisker backwards and forwards, and this is the behavior that I've been quantifying after each experiment. If anything's not clear, by the way, please stop me. Okay, so I've been doing wholesale recordings, and probably most of you are aware of this, but they're basically blind. So I'm going in the brain and pushing down through the brain until I hit onto a neuron, and then I suck onto the cell and then go wholesale. And all of my pipettes are filled with a dye, and using this technique, I obviously record the intracellular membrane potential of the neurons. These are 2 neurons that I've stained during an experiment, and these are sort of typical cells that I'm going to show you I've recorded from in these experiments. These are the layer 2-3 pyramidal cells, excitatory neurons. Here I'm recording from the soma. These are all the dendrites and the axon, and this is the barrel here of the C2 barrel column in layer 4. Okay, so what does a wholesale recording from this cortical layer look like in an awake mouse? Here's a typical recording. So this is the whisker movement. Here we've got protraction is up and retraction is down. This I've tracked after the experiment. This is a period of whisking. So he's just spontaneously started to move the whiskers backwards and forwards because he's interested in something maybe, but he's whisking in air, so there's no objects in the way, it's just moving through air. This is the membrane potential of the neuron, and you can see these large, slow oscillations here when the mouse is not moving the whisker, and then when he starts to move the whisker, we see the large, slow oscillations are reduced in amplitude, and we move to a period of high frequency, low amplitude oscillations, very reminiscent of this Berger effect that we saw in humans. So on top of this large depolarization here of the membrane potential, you can see there's small fluctuations of the membrane potential. And if you average from the peak here of each of these protractions and take an average, you can see that the membrane potential is oscillating in phase with the whisker position. So this is very small. It's only 2 millivolts or 3 millivolts. And the phase of this oscillation varies from neuron to neuron. But within one cell, the peak here is always very reliably at a certain phase of the whisker position. And we think this might be important in sensory coding. But more about that later. So these small oscillations are here riding on top of this global depolarization. I went on to make some sort of some combined recordings. So I used the wholesale technique here. This is a neuron that I've stained. This is the recording here. Then I've got an LFP electrode, which is just my wholesale electrode filled with extracellular solution, and that's just stuck in the brain here. And then I've got an EEG wire under the surface of the skull. And the EEG wire records the global activity of many neurons. The LFP is a bit more focal, the local field potential recording the activity of some cells around here. And then I've also got my intracellular wholesale recording. So as you can see here, you get the large, slow oscillations when the whiskers are still, and then there's this change in so-called brain state when the whiskers start to move. And this is reflected in the LFP as well. So this LFP has been inverted, in case you're interested, but the main point is that you also see this change in brain state in the LFP, and to some extent in the EEG, although the EEG recordings are not so pretty, I would say. The main point is that there's a change in brain state in all these recording configurations. Something that I haven't published but noticed, this sort of preliminary data, is that in fact you see these changes in different cortical regions, not just in barrel cortex. So in fact over here in visual regions you also get this change. In motor cortex you also see this going from large slow oscillations to faster membrane potential oscillations, and also in other somatosensory regions outside barrel cortex. So it appears it's a global activation of cortex when the mouse starts to move his whiskers. a common theme that I read about when reading about these brain state changes was that the neurons became desynchronized during this active state. So I just drew a little cartoon here just to help me. And what do I mean? Well, okay, the EEG is a sort of sum of all these activity of all these cells lying underneath. You can see here perhaps in this state here, they're all oscillating with quite large amplitude, and this generates a large amplitude EEG oscillation. But then you could imagine people thought, okay, well, the reason we're seeing the reduction in this amplitude is because the neurons start oscillating out of phase with each other. And so the EEG signal is just reduced in amplitude. But of course, you could imagine there were different things going on. You could imagine that, in fact, they are completely still in phase with each other, but they're just reduced in amplitude, or they're both. There's a reduction in amplitude of the signal in the individual cells. You could imagine a lot of different possibilities. So to test this, I made dual recordings from nearby cells, doing the same thing, whole cell recordings, but from cells that were very close to each other, and then correlated the activity in the cells to see whether they really were desynchronized during this active state. Here's a typical recording of, this is the two neurons that I stained. Here's the recordings from them. And you can see that they're really oscillating very much in phase with each other during these quiet periods when the whiskers are not moving. And then when the whiskers start moving, we see the change in brain state. This is the cross-correlation with this large peak from a period of quiet here. So very highly correlated. And indeed, when the mice start to move the whiskers, there's a reduction in the correlation. And this was very robust across all the pairs of neurons that I recorded. They're still correlated to some extent. This is not zero, but there's a significant reduction here. So they really are becoming desynchronized to some extent. This is just another example. And in fact, I think I have this one as a movie. Sorry, I don't know how that... Okay, there we go. Okay, so this is slowed down a number of times. You can see here they're oscillating in phase with each other. Mouse is moving his whisker. I'm tracking actually this one, and that's this movement here. And you can see they go from large slow oscillations here. This cell produces an action potential, which you may have noticed there weren't very many action potentials so far that we've seen, but I'll talk a bit more about that in a minute. And then during whisker movements, we get this period where the neurons are more desynchronized during whisker movements. And then when the whiskers stop moving, we go back to this larger slow oscillations. Okay, so in this experiment, this is another pair of cells that I've recorded, and in fact just done the cross-correlation on 1 second bins, and this is the peak of this cross-correlation, just to show you that when the mouse starts to move his whiskers, this cross-correlation desynchronization is very rapid. So this is the peak here. You can see as soon as he starts to move, there's this reduction in correlation of the neurons, and this lasts for the whole time. He's moving his whiskers, and then they go back to this recorrelated phase here, and the mouse is moving his whiskers. Stop moving. In some experiments, I've then introduced an object into the way of the whisker to have him actually touch an object. And here we can see a period of quiet from a pair that I've recorded, pair of neurons. And you can see they're highly correlated here. He starts to whisk in air, and we get this decorrelation of the membrane potential. And then I've put an object in the way. Here you can see the object here, and every time he touches, this is a zoom in of one of these touches here. Each bar here represents a touch, and you can see each touch causes a excitatory sensory response in both neurons. And indeed, they're very highly correlated in amplitude. So the amplitude of the response in one cell is very similar to the amplitude of the response in the other cell. If you look at the correlation, you can see here, in quiet periods, neurons are very highly correlated. Here, they're much less so. But then during these periods of sensory processing, actually the correlation increases, mainly because we've got these large synchronous excitatory events. So as I said, you've probably seen that there are not very many action potentials that we're recording. And in fact, this has been seen by a number of other labs doing imaging and extracellular methods of recording, but is quite different to the view maybe from some time ago using metal electrodes. And we think this might be because the whole cell technique isn't biased towards sampling neurons that are firing. So when we go through the brain, we hit a neuron and we record from it, whereas an extracellular technique might go through the brain and look for a cell that's firing because they can't see cells that are not firing. So we think this might in part be the result, why we see this difference in the numbers of spikes. Here I've just cut a 10-second segment from a recording. So this is 50 seconds in total, and you can see this neuron fires one spike in 50 seconds. This is not unusual. Here they're firing a bit more. In fact, the ones here, I will come back to this. And this is just to show you that some cells, in fact, do fire action potentials quite a lot, but this is quite rare in the cells that I've sampled. And here's the distribution here. The mean firing rate is around less than once per second. Cells do fire in bursts of action potentials as well. In fact, about 40% of the action potentials are in doublets or triplets. But these are even rarer, around 1 hertz. So the overall firing rate is quite low in this cortical layer. In other layers, we now think that there are more spikes. And this is something that appears to be very interesting, but I haven't addressed here yet. If we zoom in on a spike, this is a single spike here, single spontaneous action potential, and this is a burst. You can't see it's a burst, but in this next, I've zoomed in here on this one. You can see there's two spikes here. You can see before every spike, there's this large amplitude depolarization of the membrane potential. So the neurons, they don't fire very much, but when they do fire, they're triggered by very large input. If we measure it by just measuring 20 milliseconds before the spike, this so-called pre-spike depolarization, you can see that in these 20 milliseconds, the neurons are depolarizing by about 10 millivolts. So it's quite a significant amount. Interestingly, this large depolarization, if you also zoom into spikes and take an average from one cell, and then you look at the membrane potential of your nearby cell, it's not there. Even in quiet periods where the neurons are quite highly correlated, you do not see this huge depolarization in the nearby cell. And this is represented here on the population of all the pairs that I recorded. So we're not quite sure what the mechanism is of this. It may be some sort of internal cell. Yeah. Do you have a? Yeah. So here, you mean this sort of oscillation? Yeah, well, if you average across lots of spikes, I mean, also, okay, so the time scale is quite different. So here we've zoomed into 50 milliseconds on either side. So it may be that in part is I mean, if we had a much longer time scale, but on. Yeah, it's difficult to say, I guess. Yeah, I mean, certainly in some cases they appear on top of a sort of slower oscillation. In some, it just appears to be the rising phase of the start of the oscillation. So it's difficult to comment, I'd say. But yeah, we're not sure really whether this is a network phenomenon or an internal property of the cell that's triggering this. We're just not sure yet. But it's certainly quite prominent effect. This happens both during whisking and during quiet periods. So this is both, you see it both in both states. You see this big depolarization. So even when it's high depolarization during non-whisk, yeah, you get actually the type of activity which is uncorrelated. Yeah, exactly. Yeah, exactly. So it's sort of on top of this. Wow. This, yeah. If you zoom in and look at the threshold for action potentials here, so this is the threshold here which triggers the spike, we also notice that this varies quite a bit by some millivolts. And this seems to depend on the gradient of this depolarization before the spike. So this has been noted before in cats, in anesthetized cats. If there's a slow gradient here, you can see the threshold is higher, and then it's lower. So it seems that you're more likely to trigger an action potential if you have fast synchronous input to a neuron. And here we've quantified this. Okay. So, yeah, actually, I'm going quite quickly. I'm now interested in what the mechanisms are of this brain state change. And I guess the first idea was that it could be sensory feedback. So it could be that every time the mouse moves his whiskers backwards and forwards, sensory signals are picked up at the whisker pad, and these come back up the infraorbital nerve, and these change the whole state of the cortex. Or you could imagine that it's somehow internally generated. So either maybe a motor center that generates the whisker movement, or some other nucleus predicts the mouse is going to move and somehow prepares the sensory cortex for expected lots of sensory input that's just about to come because he's moving his whiskers, and it's somehow internally generated. So as a first step to tell the difference, I cut the sensory nerves here, the infraorbital nerves on both sides of the mouse, here and here, and then I made the same recording. So if the brain state change is still there, then we know that it's somehow internally generated, and if it's gone, then it's sensory. Okay, this is back to the control recording with the nerves intact, and here you can see this change in brain state, and when I cut the nerves, basically it's still there. This neuron, in fact, is firing a bit more than usual, but here you can see this change in brain state from large, slow oscillations to fast input. So this is not sensory. In fact, when we did some analysis on lots of properties of the cells during these brain state changes, here you can see I measured the power in a certain frequency band, I measured the variance of the membrane potential, the actual mean membrane potential, and the frequency of action potentials, and everything. There was no real difference between nerve intact and nerve cut, which is kind of surprising, especially when it comes to action potential firing, because it basically means that the action potentials mostly are not sensory. Even though we're in primary sensory cortex, you can remove the sensory nerve, and there's still the same numbers of action potentials. Something did change, however, and that was this small amplitude oscillation on top of the overall brain state change. And when I cut the sensory nerves, this is basically gone. So this is another protraction-triggered average here. I've averaged from this peak here and averaged the membrane potential. You can see the oscillation when the nerves are intact, but when the nerves are cut, the membrane potential is flat. So work I've been doing recently, so we know it's not sensory, and then I thought, well, perhaps the thalamus is involved. Perhaps this relay station here, or so-called relay station for sensory input, is involved in generating this brain state change. But as yet, there were really very few recordings even from the thalamus in awake animals, so we didn't really know what to expect. I made some recordings down there, and I'll show you these now. This is a typical recording, and in fact we've got a number of cells now, and they all pretty much do the same thing. And you can see the whisker movements here during a period of whisking. This is the LFP in the cortex, the barrel cortex. You can see this brain state here, this active state, and then when the whiskers stop, you can see these large slow oscillations. And this is an extracellular recording from a single cell in the thalamus that is also processing whisker information. And you can see here that these thalamic cells are firing quite a lot during whisker movements. So something is causing the thalamus to fire quite a bit. I'm not exactly sure what it is yet. But as a next step, I actually tried to inactivate this spiking. And to see how this would affect the brain state change in the cortex. So I inactivate the thalamic input and then see how this changes this change. And to do that, I had the same preparation. I had a recording in the cortex, but then instead of an electrode here recording thalamic activity, I went in with a drug, muscimol, that activates GABA receptors and silences neurons. So you inject this drug and it silences these cells. So this is the preparation. This is my muscimol injection in the thalamus, and this is my recording. This is first a local field potential recording, so this is extracellular, and here you can see this is before I've injected the muscimol. Here you can see the state change from slow oscillations to fast. And here, after I've injected muscimol, something really dramatic happened, and we weren't really expecting this. That we still had big oscillations, quiet periods, but then instead of this sort of state change that appeared to be lots of synaptic input, there was just a flat line. And in fact, I went on to make whole cell recordings instead of just extracellular recordings, and we saw it in whole cell recordings as well. So this is a control recording, and then you can see after I've inactivated the thalamus, it's very weird. There's basically no synaptic input at all during the period of whisking here once I've inactivated the thalamus. So it appears I removed this excitation from the thalamus, and this completely removes this depolarization here. There's just only a few EPSPs you can see here, and the membrane potential is just completely flat. There are still some slow oscillations. They reduced in frequency, so there's less of them than in the awake state, and they're sort of bigger, and they represent more, they're more like an anesthetized preparation, so you see these large slow oscillations. Still, some questions remain, okay? So we think the thalamus is really heavily regulating this, but what is triggering the thalamus to fire? I'm not sure, but I'm thinking perhaps it's neuromodulators, which is why I'm sort of interested in neuromodulators at the moment. It may not be, it might be other things. But also, even once we've removed it, we still see this shutdown here. We still see some sort of change. So what's causing this? Why is it still there's some sort of change? Okay, it's not the normal depolarization with some spikes and synaptic input, but there's a complete shutdown of the slow oscillations. And again, I'm sort of interested in what's the mechanism underlying this. So I don't know yet, so maybe I'll tell you in some years. Yeah, so I've actually gone quite quickly. But so in conclusion, we've got the layer 2-3 cortical neurons undergo this change in brain state during whisker movements. Neuronal activity is very correlated when the mice are sitting still, but when they start to move their whiskers, the neurons desynchronize and start to do different things. Action potentials don't happen very often in awake mice in this layer, but are listed by very large amplitude inputs, and they seem to be cell-specific. So one cell will fire and the other cell will just not at all. The state change is certainly internally generated. The thalamus is heavily involved in regulating this state change, and we think that overall it might increase the sort of information capacity of the brain. Just at the time the mouse is trying to build up a picture of the world, it might somehow allow, because the neurons start to do different things. You could imagine if they're all doing the same thing, it might be difficult to code for lots of different sensory input. But with this desynchronization, we imagine that this might improve the overall information coding capacity. Yeah, so thanks a lot for listening, and if you've got any questions, please feel free. And thanks to Carl, who was my supervisor for all this work, and the members of this lab. And I was funded by the Human Frontiers and also the Swiss National Science Foundation, and some money from Roche. So thanks very much.